Okay, in this video I'll be talking about alternative splicing, which is the ability of a cell to splice an mRNA transcript in different ways to create totally different polypeptide chains. And in my little diagram, the little lightly shaded areas, those are my exons. The lightly shaded yellow areas, those are going to be the introns. And the darkly shaded yellow areas, those are the exons for only one of the mRNA transcripts, or for one of the mRNAs. And these little red triangles you see throughout the diagram, those indicate where an intron is going to be removed. The first example we're going to be going over is exon skipping, and as you can see here, the number 1 line, that side of the mRNA has skipped that section of exon, so that one won't. So that little section of exon won't be included in the final protein. And the second one shows intron retention, and basically this means that you can see that the bottom one doesn't excise that certain section of mRNA, so it's going to keep that section and include it in the protein. Thirdly, we have alternative 5' splice site, and what happens here is you can see on the bottom one that it's going to express just a little bit more of that DNA as opposed to the top one, so it's going to just express just a little bit different of a protein. Next, we've got alternative 3' splice site, which is basically the same thing as alternative 5', but now in the other way. And lastly, we've got mutually exclusive exons, and basically what this means is that the top one is going to excise an intron that the bottom one is going to include as an exon, and vice versa. And as you can imagine, this is a very, very, very powerful tool that our bodies are able to use in order to create a bunch of different proteins from the same genes, up to like I read 38,000 different proteins from a single gene.